So the first item would be the Xray Diffractometer. It's an extremely powerful tool for material characterization. It is widely used for identification of unknown crystalline materials. Say crystalline materials, solid sila ng mga materials, not liquid. So, for example, kay unknown material na solid, basta solid siya, you have to ground it first para makita siya because we are scanning a molecular level ng item. So that's why it has to be extremely minute. I will show a video sa iyahang methods sa pag set up sa machine.
What is X-ray diffraction, and what is it used for? In an X-ray diffraction experiment, a sample is placed into the center of an instrument and illuminated with a beam of X-rays. The X-ray tube and detector move in a synchronized motion. The signal coming from the sample is recorded and graphed, where peaks are observed related to the atomic structure of the sample. Most materials are made up of many small crystals, like sand on a beach. Each of these crystals is composed of a regular arrangement of atoms, and each atom is composed of a nucleus surrounded by a cloud of electrons. It's at this scale that the story of X-ray diffraction begins. X-rays are high-energy light with a repeating period called the wavelength. Since the wavelength of an X-ray is similar to the distance between atoms in a crystal, a special interference effect called diffraction can be used to measure the distance between the atoms.
Upon entering the XRD room, the user should take notice of the operational lights on the side of the instrument. The ready and on lights should be illuminated, while the alarm and busy lights should be off. To unlock the doors, the user presses the open door button. The alarm light will blink while the doors remain unlocked. To open the doors, the user pulls the two black handles towards themselves simultaneously, then gently slides the doors apart by the metal fixtures. X-rays are generated by accelerating electrons into a copper target. X-rays are directed towards the sample where they diffract. Angles of constructive interference will diffract X-rays into the one-dimensional detector, which transduces the X-ray intensity. The X-ray beam leaving the X-ray generator is confined by the divergent slit. The length of the sample that is illuminated by X-rays depends on the diffraction angle and divergent slit size. A narrower divergent slit produces sharper diffraction peaks at lower intensity that can help resolve closely spaced peaks. On the detector side, there is an anti-scatter slit which reduces noise caused by scattered X-rays from amorphous material or air. The diffraction slit further controls the resolution of the instrument. In between the two slits is a piece of nickel foil. Nickel has an X-ray absorption edge between copper K-alpha and K-beta, causing it to absorb more of the higher energy K-beta X-rays while letting more of the K-alpha X-rays pass through it. There are various types of sample holders for different samples. No matter what type of holder is used, it is important that the surface of the sample is flush with the top surface of the sample holder. This sample holder has an adjustable stage to change the height of the sample. After loading a sample, the sample holder is placed onto the base of the sample stage fixture. The sample holder is lifted up from above so that its top surface is flush with the top of the sample stage. Then, the sample stage fixture is gently pressed up, locking the sample holder into position. The doors are closed by gently sliding them together by the metal fixtures, and then locked by pressing the black handles in simultaneously. When the doors are locked, the alarm light will stop blinking and remain off. A scan method can be created or modified using the XRD Wizard application from the desktop. From the application screen, a new method or previous method can be opened. The user can set the 2 theta scan range, step size or number of steps per scan, and time per step. The X-ray generator power for the scan can be adjusted up to 40 kV and 40 milliamps. Finally, the scan method is saved. Operation of the instrument is done through the XRD Commander application. Before starting a scan, the X-ray generator power must be slowly ramped up to its desired output. First, voltage is increased at 5 kV increments by typing in the next value and clicking the set button. The X-ray generator is allowed to rest for 10 seconds before the user increases to the next set point. This slow ramp in power will ensure increased longevity of the X-ray generator. Next, the current is increased at 5 mA intervals in the same manner. After the target power has been achieved, the user can set up the next scan by clicking on the Jobs drop-down menu and selecting Jobs. This will open a new window where the user can add a sample ID, select a parameter file, and save location for their sample. The user starts the selected scan by pressing the Start button. This will open the shutter of the X-ray generator and begin the scan. 2 red lights are illuminated on the X-ray generator when the shutter is open, and the goniometer moves into its starting theta and 2 theta positions. This goniometer has a stationary X-ray source with moving sample stage and detector, but other XRD instruments may have a stationary sample stage with moving X-ray source and detector. During a scan, the busy light will be illuminated. When the scan has completed, the X-ray generator shutter will automatically close and the busy light will turn off. Now the sample is ready to be removed. The final position of the goniometer may make it difficult to remove the sample holder. The user can adjust the goniometer position manually by changing the theta and 2 theta values to 30 and 60 degrees and pressing the Move Drives button in the XRD Commander application. To remove the sample holder from the sample stage, a latch underneath the sample stage is gently pulled while holding onto the sample holder. Before leaving, the status of the alarm light is checked. If the alarm is on, the user will contact the lab technician for assistance.
The next item would be the differential scanning calorimeter. This item measures temperatures and heat flows associated with thermal transitions in a material. And when they use this differential scanning calorimetry technique, polymers are observed and studied what happens when they're heated. So basically, they're saying that the main use for this item is para ma-measure ang activity sa mga polymers pag mainitan sila, like enzymes or any other inorganic materials or organic man. And when they're heated, ay activity nga mahitabo pag mainitan lang sila or pag ma-denatured sila. And that's why scientists need to know those temperature threshold para pag need nila mag-elicit og certain activity sa polymer or enzymes, they will have the knowledge kung kanusa sila ma-denature or mag-exert og certain activities pag mainitan. Those thermal activity nga ginastorya nako is what we call the thermal transitions. Muna ang heat nga maitabo sa mga polymers pag imo silang mainitan. And so the first step para maimong mastudihan is to heat those polymers, of course. So when you heat a polymer, they release some kind of exothermic energy. When they release those heats, sooner or later they will reach a point nga they will break or be denatured. Like the enzymes, and the point of it is to observe their activities while they're heated and when they are denatured. It's an extremely useful item to observe the oxidation of other polymers and other chemical reactions. Dili lang ang ilahang heat measurement at ilahang denaturation points.
What is DSC? Differential scanning calorimetry, or DSC, measures the heat change during thermal denaturation of biomolecule samples, particularly engineered proteins and biopharmaceutical products. DSC is universally considered the gold standard for measuring TM, or the melting temperature of proteins in solution. DSC presents the TM as the peak of the data plot. Importantly, DSC is the only known reliable method to directly measure unfolding thermodynamics of protein samples, from a native active conformation to a denatured inactive conformation. DSC thermal stability assays provide details on how the protein unfolds, enabling protein engineers to identify stable constructs, as well as optimizing purification strategies and storage conditions. The main parameter measured by thermal stability assays is the thermal transition temperature, or TM. The TM is the temperature at which 50% of the protein is native, and 50% is denatured. It's the most reliable indicator of thermal stability. The higher the TM, the more stable the protein is, which is why TM screening is universally used to select the most stable engineered protein, as well as the buffers and additives which can further stabilize it. Reliability of technique. DSC is label-free, using only native molecules. This eliminates the need for added dyes or chemical modifications, producing more reliable results than traditional fluorescence assays, which frequently introduce unwanted artifacts. Using DSC equipment. Malvern's MicroCal Peak DSC systems use small sample volumes, are easy to operate, can also be automated for high throughput with solutions available for 21 CFR Part 11 compliance. MicroCal DSC data is the trusted gold standard. For more information on MicroCal DSC and how it can support your research, please visit malvern.com/microcal or contact your regional Malvern sales office.
Colorimeter, or the automatic colorimeter, involves the measurement of color and is widely used for method finding the concentration of biochemical compounds. Mao ni siyang item nga katandem sa spectrophotometer. Ang kalayan ng atong spectrophotometer is it also measures color and light, while the colorimeter just measures the color of a certain concentration substance. It measures the absorbance wavelength between 400 to 700 nanometer from the visible spectrum of light of the electromagnetic spectrum. So kaning 400 to 700 nanometer, mura ni sila ang visible spectrum of light kanang makita ra sa mata, visible lang, naked eye. So if namoy mga studies about compounds like phytochemical screening, there's some kinds of compounds na kailangan ma-determine ilang concentrations and solutions. So scientists would use these methods colorimetry or spectral photometry para ma-determine ang absorbance rate, ang concentration sa certain biochemical compounds. Colorimeters are important because they provide reliable and highly accurate results. For example, mag-assess ka color sa bird sa ilang feathers, or pag mag-measure ka o mag-monitor ka sa color sa various foods and beverages like anything, and including vegetables, sugars, screening the quality of water, quality of your concentration sa imong compounds, sa imong thesis pananglitan, sa imong plants. Na ba sila any impurities when they are so precise, molecular level.
Inside the colorimeter, a light source is directed at a diffraction grating that resolves the light into its constituent wavelengths. By turning a dial, one is able to select a narrow range of wavelengths to shine through the sample. That typically is the complement of the color of the sample. In this way, the maximum amount of light will be absorbed. Turning the dial rotates a cam that causes an arm to twist the position of the grating. This allows a narrow range of wavelengths of light to pass through a slit and into the sample. Some or all of this light is absorbed, and the rest is transmitted to a light-measuring photoelectric tube that causes a response in a meter. To run an analysis, we turn on the instrument to allow it to warm up for at least 15 minutes. Next, we use the top knob to select the proper wavelength of maximum absorbance. One test tube that we'll call the cuvette is used for all measurement. This will minimize errors due to differences in the glass between different test tubes. First, the blank, or control, that contains all chemicals except the light-absorbing species, is poured into the cuvette, which is then inserted into the sample holder of the instrument. Note that the test tube's label is oriented to the front of the sample holder. We turn the 100% adjust dial so that the needle is set on 0 absorbance, or 100% transmittance. Now we remove the cuvette from the instrument and close the top flap, and use the dual-purpose on-off knob to select 0 transmittance, or infinite absorbance. We repeat this process until the meter does not drift. We get 100% transmittance with the control cuvette inserted and 0% transmittance with no cuvette in the instrument. We then pour the control solution back into its own test tube, rinse out the cuvette, and add the lightest colored sample to the cuvette after rinsing the cuvette with a small amount of the sample. We insert the cuvette into the instrument, always with the test tube's label pointed toward the front of the sample holder, close the top flap and record both the transmittance and the absorbance. We pour the sample back into its test tube and rinse out the cuvette with distilled water. We now rinse the cuvette with a small amount of the next darker sample, then fill the cuvette with it and determine its transmittance and absorbance. After the second reading, it is wise to use the control solution again to recheck that the instrument still reads 100% transmittance with the control solution in place and 0% transmittance with the sample holder empty. Continue taking readings of all of the solutions of known concentration, then run your unknowns. Do all of these readings at the same time using the same colorimeter, periodically checking the instrument with the control solution. After you have collected all of your data, you can transfer the information to a spreadsheet program to graph the absorbance on the y-axis versus the concentration of the species being analyzed on the x-axis. Instrumental methods are profoundly important in chemistry, and we hope that you find that your experiences with colorimetry will serve as an interesting introduction to this type of analysis.
Microbiological Incubator, or the cell culture incubator, is an insulated and closed device that provides an optimal condition of temperature, humidity, and other environmental conditions required for the growth of organisms. They are widely used for culturing of cells, both eukaryotic and prokaryotic. Having a farm of instead chickens and cows, you're going to have a farm of microorganisms like viruses, bacteria, fungi, and this machine sets up an ideal environment for your organisms to thrive. Microinjection pipette uses glass microinjection needles with very fine tip, around 0.1 to 0.5 micrometers, to directly inject foreign gene fragments into pronuclear embryo or cultured cells in America through the host genome sequence. And around 1911 by Dr. Marshall A. Barber, he proposed this new technique called the microinjection technique. He invented this microinjection to initially clone bacteria and to confirm the germ theory of Koch and Pasteur. Nowadays, it is common to use this for using recombinant DNA technique, or the insertion of insemination egg obtained from embryonic donor's fallopian tube to the inverted microscope. And in case if worried mo kung giyon sa nila pagbuhat aning super thin gamay nga needle, is they use the capillary tube and slice it by a machine or manually slice it.